Brothers and sisters, Assalamu alaikum. Thank you all for coming. Whether you're here with us live on Zoom, live on YouTube, or whether you're watching this after the event, I would like to welcome you all to this evening's discussion. We find ourselves in a rather uncertain situation as we approach Christmas in the year 2020. It has been a year like no other. We have changed the way we live. We fear for our lives when we leave our houses and refrain from seeing our loved ones as we are concerned about their welfare. We've stopped going to school, going to work, going to the mosques, and even think twice before going to the supermarkets. As a country, the UK has officially had 1,723,242 cases of coronavirus confirmed, and has had at least 61,242 people die because of this disease. And if we look at excess deaths in the year 2020, the numbers are even more concerning. Not only is staying at home ruining people's mental health, but as a result, the UK is forecasted to go into recession in the early part of 2021. Major businesses are going bankrupt and unemployment rates are sky high. We are looking at a situation where case numbers go down only during a lockdown, but as soon as we try and reopen, cases start climbing, pushing us into a further lockdown. But finally, there may just be a light at the end of the tunnel. From Tuesday, the UK will start its vaccination program. The vaccine has been hyped to be a get out of jail card. We are told that this is what will finally bring us back to a sense of normality. But the vaccine raises, in my mind at least, more questions than it answers. Is it safe? Has it gone through the necessary testing stages? Does it work? To help give us a better understanding of things and answer some of these questions, we are very fortunate to have Dr. Umar Ebrahimsa to talk to us. Dr. Ebrahimsa is a specialist registrar at the Royal London and has spent the majority of 2020 working in COVID wards. He specializes in microbiology, infectious diseases, general medicine, and virology. I'm sure people will have loads of questions to ask Umar. So if you place your questions on the chat, either in Zoom or on YouTube, I will try and put forward as many of the questions as possible after the talk. So without further ado, I hand over to Dr. Ebrahimsa to deliver his talk.
Assalamu alaikum. Thank you very much for that kind introduction, Mohammed. And as you mentioned, we are living through, as they say, unprecedented times. And it's humbling to think that very few of us would have anticipated ending up in the situation that we're in today, looking at what might have come at the end of 2019. But alhamdulillah. Alhamdulillah wa salatu wa salam ala rasulillah. Assalamu alaikum. As Mohammed mentioned, I'm a registrar in infectious diseases and have spent most of this year working in one form or another with patients or laboratories on COVID-19. I speak in my personal capacity, but my day job is based in East London, which has some of the highest incidence rates of COVID-19 within London and the UK. What I wanted to sketch out today was a route for us to understand the possibilities opened up and the challenges posed by the recent announcements and the recent deployment of vaccines against coronavirus, and how we can approach this and how we can make reasonable decisions at the end of this. And I start off all of this looking back at a year where 60,000 people in the UK have died. At a year where, as Mohammed mentioned, we've had mosques and churches, synagogues and gurdwaras closed and emptied, where the other day I met someone who said they hadn't been out of their house for a good 6 months except for short breaks, where life itself has been turned on its head. And where the impact of this virus has cut to the very, very soul of what it means to be a human being, how we relate to one another, how we meet one another, how we greet one another. And that's both worrying and humbling. But how do we begin to understand the news a few days ago that the UK had authorized the purchase and then subsequently the distribution of vaccines produced by Pfizer BioNTech against COVID-19? Well, I thought it may be useful to take us on a journey, not to the stars, not on some wonderful moonshot, but to the very heart of ourselves, to the very bits of us that define us, define the materiality of ourselves. The cell. Imagine, if you will, that you're sat on your chair, surrounded by this pool of living jelly, this substance that defines who you are. Swimming around in it are the things that define what you pass on to your children, are the things that determine how well you live within this world. And around it all is that impenetrable great wall of the cell membrane, punctured by archways through which things can pass in and out. A mysterious layer that can accept things passing through it like ghosts, and at the same time prevent others from moving anywhere near it. And over there in the corner is this great mass, again surrounded by another membrane with huge portals within it, hiding your genetic material. This great library of codes, coding for the some 20,000 proteins that make us up, all joined together, tightly wound in a rope that is wound upon itself, that binds together to form the chromosome. And as they sit there, your body, when it needs, or your cell, when it needs to produce a protein, can just copy one of these into a stream of RNA that you see here, just running across past me. And over there in the corner, these factories, these ribosomes accepting this train of RNA, this message that's come out of the nucleus, coding for a protein. And then over on that little lacuna, in that space like an airship, a whole host of proteins produced by the ribosome. So what is it that RNA does? It transfers information from the nucleus, from the very thing that passes from mother to daughter. It transfers it out and enables it to become useful and make the proteins that we need. So hold that thought for a moment. And let's look at something else. The whole debate over the last few days has been about vaccination. But what is vaccination? And hold in your mind's eye this image of a man traipsing from village to village, surrounded by dust, clothed in just white cotton, with his stick forming the path, and in his knapsack, the very core of what we today see as inoculation, immunization, the means of protecting yourself against disease. This could quite easily be a scene anywhere from Turkey to India to West Africa, but not significantly something that you saw within London or the darker climes of Western Europe. For the inoculator was the man who protected societies against smallpox, infecting individuals with a weakened version in order to protect them from the greater harm that smallpox threatened them with. And that essentially is what we're doing with the vaccine. We're trying to get the immune system, the thing that defines self and other, to get it to produce a response against something that we want it to see as other. So imagine again, of those 20,000 proteins that we have, most are contained within the cell. But there are some that line the pathways of our bloodstream, the surrounding of our nerves, the surface of our skin, all areas which our immune system targets, that surveys, perpetually alert, looking out for a threat. And the threat that it identifies is a protein that it doesn't recognize. So somehow it's able to protect itself from attacking itself, to prevent itself from recognizing those 20,000 as being foreign. Rather, not foreign, being a danger. But what we're doing is getting it to recognize this particular protein as being a danger and to respond to it. And the way the immune system does this is to produce an unveiling gel, a shower of little molecules that fall upon the protein and highlight it, like dust that then shines and declares that danger coming towards you. And these are your antibodies. But your antibodies are produced by one set of your immune cells. And then on the other side are the cells that come in and eat up the little Pac-Man of our immune system, swallowing whole all of these bacteria and viruses and cells infected by them. And what we want to do is to alert the immune system, to encourage it to produce antibodies, to activate those Pac-Man. And the way you do that is to introduce this protein to it. And there are several ways we can do that. For a virus, you could get a dead virus and show it to the immune system. Or you could get a weakened virus, as that inoculator was doing with smallpox, and show it to the immune system, and as we do today with polio. Or you could get a bit of that virus, mash it up into small bits, and then tie it together with another protein in order to encourage a response and display it to the immune system. And we do that with some of the vaccines that we have currently, like measles. Or what you can try and do is get the body itself to produce these proteins. And that is what's happening with these 3 new vaccines that we have against coronavirus. The one produced by Pfizer-BioNTech and Moderna are very similar in the sense that they are that mRNA that we saw swimming through the cell, wandering out from the nucleus and going off to the ribosome as it produces the protein. And this is a bit of mRNA that codes for the spike protein. So let me paint you another picture of a ball with little rods coming off it and cups at the end of those rods, spikes that spear our cells, that hold on to them, that allow the virus to enter into us. That is a spike protein, one of several that surround the coronavirus. and it seems probably the most important in allowing entry of the virus into us, and the target of most of the vaccines that are being developed. So Pfizer-BioNTech and Moderna have produced a section of mRNA that codes for this. And they try and get it into the cell. Now, remember, we talked about that impenetrable barrier on the edge of the cell, that great wall of the cell membrane, which could only be entered through certain portals. Well, here, this vaccine acts almost like a ghost. It comes, it's swallowed by that wall, it swims through it, and then emerges within the cell. And it's able to do this because it's a droplet of fat with the mRNA hidden within it. So the mRNA is not exposed to the immune system on its way into the cell, but it enters, it swims through the cytoplasm and ends up being used by your ribosome. And because it is an mRNA, it is this intermediate step. It can't reproduce. It's only there for a short while. It produces a protein and it's never, ever integrated into that hub of the nucleus. And once that protein is produced, because it's so foreign, it's then also dangerous to the body. It then comes on to the surface and activates your immune response. And that essentially is how both the Pfizer and the Moderna vaccines work. The Oxford vaccine, on the other hand, is slightly different in the sense that it is a virus, but a virus that can't reproduce. It's a non-replicating virus. And it's a virus that causes the common cold. What we thought, let me rephrase that, one of a family called the adenoviruses. But it's an adenovirus that affects not humans, but monkeys, so that you don't have an immune response to it. And it comes up to the cell, injects a bit of DNA into the cell. But because it doesn't reproduce, this DNA, all it does is produce more and more mRNA, which is then used to produce the protein, used to produce the spike protein. This then, like the Pfizer-BioNTech and the Moderna vaccines, this protein is then brought up to the surface, displayed, and your immune system responds to it. That essentially is how these vaccines work. So they're not capable of reproducing, they're not capable of persisting, and what they essentially do is create this vast pool of proteins in the body that suddenly emerge and your immune system recognizes as being new and dangerous and responds to. All of these vaccines require what's known as a prime boost mechanism. So they require an initial exposure where you're injected, and then in some cases injected about a month afterwards, in some 2 weeks afterwards, with a second dose. And the idea of that second dose is that it augments the initial response. So your immune system initially recognizes the protein, and then when it recognizes it again, it comes at it and doubles down and causes a more pronounced response to it. And hopefully a more persistent one. So that, I think, paints a picture of what the vaccine candidates, as we know them, how they work, and perhaps how they cause side effects. So if you're going to cause an immune response, what are you doing? You're encouraging a fever. You will have inevitably feverishness, muscle aches and pains, headaches, swelling at the site that the vaccine itself is injected. So all of these are expected with any of these vaccines, just as we expect them with childhood vaccines. And then subsequently, you'd hope that there is some kind of immune response to them. Now, the best way to identify whether there has been an immune response is actually to see if people are protected by this if they don't develop coronavirus. And the question of how we define the development of coronavirus is an interesting one. I'll touch on that in the question and answers. But what I wanted to do now was just to work through each of the vaccines themselves and what we know of them so far from the trial data that's there. One of the more concerning elements this year has been the rapidity with which all of this has been achieved. And I started off thinking about this talk, wondering how it was that we were able to, within a matter of a year, in fact, less than a year, from the time we knew that the genetic code of coronavirus, from that time till today, to have a vaccine that you can actually distribute in a population, I wondered how that was possible. And very early on, there's already been chatter or discussion and worry about whether these candidates are actually all they're cracked out to be. And I shared those concerns as well. And I was worried about the other element in this, which has been the willingness to develop this, to disseminate this information, not through conventional means by publishing within a scientific journal, distributing the evidence and allowing people to critique it, but through press releases with selected data being released. However, I think we need to set that aside, the processes from the actual vaccine itself. Because looking back, both the Oxford group and Moderna have actually published their phase 2 and 3 trial data on this. But you might ask, what is this phase 2 and 3 data? What is phase 2 and 3? Well, any vaccine that undergoes development, you start off at the drawing board with a blue sky concept, let's say, and look back into the books and wonder whether this theory might actually work, whether there's a basis for it, and you try and innovate around that. Then you have a process of going into the laboratory and testing that idea out. Does it work? Does it work theoretically in the test tube, on a petri dish, or in animals? And finally, when you realize that it might work in animals, you then start looking at healthy human volunteers. And in less than 100 persons or so, you have your phase 1 trial, where you assess the safety of the vaccine. So it's not whether it works, but whether it's safe and whether we can safely use it within a human population. You then have, once that is completed, a phase 2 process, where you look at whether the vaccine works across a number of environments or different people, whether it's consistent and whether it generates an immune response. And also look at the side effects. This is in about a few hundred people. You then go to phase 3 studies, where you're interested in whether it's effective, whether there is some efficacy for it, whether it protects you from disease, whether it prevents disease occurring, and also further evidence of whether it's safe or not. And you look for the rarer side effects. And in this, you're looking at thousands of people being vaccinated. And it's at that point, you then use that data to come up to the licensing authorities, whether it's the European Medicines Agency or the MHRA in the UK from the end of the year. And even after this, you're interested in side effects. You're interested in whether there is idiosyncratic or very low frequency side effects that occur. And that's part of the post-marketing surveillance, the phase 4 of all of this. So where have our candidate vaccines? The Oxford vaccine, the BioNTech vaccine, and the Moderna vaccine, where have all of these got up to? Well, they've all actually had an extensive theoretical development. They've all been tested in the laboratory. The Oxford vaccine has been around in this form as chimp adenovirus. So the vehicle, if you like, has been used in other conditions, in other trials for over a decade. Whilst Moderna has been looking at using mRNA to treat rare conditions like Crigler-Najjar, conditions where there's an abnormality of metabolism, for, again, at least 4 or 5 years. So they've been working on this for a while, but in a different field. Just as BioNTech in Germany has been looking at this as a means of cancer therapeutics, how to introduce the mRNA into the cell. So both Moderna and BioNTech have been using mRNA and trying to get it into the cell, but for different purposes. So once the coronavirus crisis hit, they were able just to shift slightly. Instead of using an mRNA to correct an abnormality of metabolism or to correct a genetic abnormality that causes cancer, or rather not to correct a genetic abnormality, but to treat a cancer, they've looked at introducing a means of producing a viral protein to generate an immune response. So these are existing platforms that have been repurposed at speed, given the huge investments that have been put into this by government and the private sector. And all of them have gone through a phase 1, phase 2, and phase 3 study, with publication by Oxford of its phase 2, phase 3 data from the UK in The Lancet, and Moderna in some of its phase 2, phase 3 data from the US in the New England Journal of Medicine. So an element of this is available within the scientific press, and elements that we can look at to identify what are the reactions that it causes, what's the reactogenicity of this vaccine, and what does it produce an immune response, and subsequently the last bit in that chain, how effective it is, the efficacy. That element hasn't been published in the scientific press, and that's what we're dependent upon the press releases for. But I'm reasonably convinced that all the normal steps that are undertaken in order to carry out complete vaccines development have been carried out. And it's not a matter of just randomly carrying out trials at a whim. And these are massive undertakings with huge amounts of resource devoted to identifying even the slightest adverse events that occur. So, for instance, in the Oxford group and the phase 2-3 data, all of the participants were given diary cards for 7 days to look at whether they developed pain or tenderness, any problems where they were injected, or any fever, malaise, muscle aches, pains, fatigue, nausea, and diary cards in order to make sure that they don't forget and to document all of this effectively. And then on top of that, followed up after a week, 14 days, and 1 month after each of the 2 injections that they had. So that's what, for 8 points at which they're followed up. Now, when you're doing this across thousands of people, it gives a kind of confidence if there is this process in play and there's this level of documentation. But it is, as I said, unfortunate that in the final bit, that final element hasn't been released as we would normally expect it to be. So, from the phase 2 and 3 studies, so this is where, as we said, you're looking at the consistency of the vaccine's activity, whether there's an immune response, and the side effects in the main, because the efficacy data is not released in that, in several thousand people. In the case of Oxford with the chimp adenovirus, you have had publication of a study in the Lancet looking at vaccination in age groups between 18 and 70, where the adenovirus was given to one group and the control group was the meningitis ACWY vaccine, with vaccine given at day 0 and then 1 month later. And what's interesting is that if you look at this group, people get side effects, immediate side effects with the local side effects with the vaccination, with the injection site pain and tenderness, but these tend to last only about 2 days. And in terms of systemic side effects, fatigue, headache, feverishness, and myalgia, again, lasting between 2 and 3 days at most, seem to be the most common within them. So that's what we would expect within the wider rollout as well. And they all produce a good response in terms of the antibodies to that spike protein, and also antibodies that kill off or prevent virus infecting cells when it's tested in cells. So we know they're working, if you like. And the Oxford and AstraZeneca group claims in its press release, combining data from the UK and Brazil with ongoing trials in the US, where they've looked at 11,636 people, that they have an efficacy overall of about 70%. That is, 70% of people who had the vaccine were protected or did not develop COVID. And in this group, at least in the UK, I'm not sure about Brazil, but in the UK, they define the development of COVID as having symptoms consistent with COVID and having a PCR positive. Likewise in Brazil, but the UK, in addition, seems to have had weekly surveillance of everyone who's there, so they would have picked up asymptomatic cases as well. However, they haven't told us what the sub-analysis for the UK is here, but 70% overall efficacy there, with an unexpected higher efficacy of 90% in a group which received a lower dose of 2,000, a lower dose in about 2,741 persons under the age of 55, all of which are claimed to be very significant. And overall, 131 people had COVID, but we don't know how that's distributed between those who received the vaccine and those who didn't. What does this tell us? It tells us that this is a very good vaccine, but that there is some unexplained effect where having a smaller dose of virus infect you initially produces a stronger response. At least this is what the data immediately says. What does it suggest? Probably useful as a vaccine which can be stored within a fridge for at least 6 months, widely rolled out, much cheaper than the others that are there, and with evidence of good response in persons above the age of 65. Now, it would be useful when the data comes out for us to revisit this and to see what the differences are between the UK and Brazil, and to see just how diverse and varied the study cohorts are in terms of gender, in terms of ethnicity. They don't assess for this, but in future, actually, in terms of socioeconomic, some marker of socioeconomic class. Moving on to the other two vaccines. So this is the viral vector vaccine that we're looking at. The other two vaccines are the mRNA vaccines. So these are the lipid layers with mRNA within them that fuse with that cell membrane and then release it into the cell to produce the protein that the immune system will respond to. And two companies have produced these vaccines. One is Pfizer-BioNTech, and In the press release, they state that they have enrolled 43,000 people in their trials. So 43,000 people, of whom at least 41,000 have had 2 doses, the first vaccine and the second one. And out of the cohort they've looked at, 170 people have developed COVID, 162 in the control group, so the people who didn't get the vaccine, and 8 in those who had the vaccine. And they claim an efficacy of 95%, with an efficacy of 94% in those who are above the age of 65. And in terms of the side effects of this, no one has had to be hospitalized as a result of the vaccination. No one has had severe illness as a result of the vaccination. The most common side effect has been fatigue and headache, with about 3.8% of people having fatigue after the vaccine and about 2% having headache. All other side effects occur in less than 2% of persons. So again, not particularly worrying from a side effect perspective. And then we come to the Moderna vaccine. So this, the press release is probably the most detailed, and again, as I said, it's from the press release is probably the most detailed of the 3 that have come across the table. They had a cohort of 30,000 people. So how many do we have? So 30,000 in Moderna, 40-odd thousand in Pfizer, that's 70,000 so far. Oxford haven't told us the total number, so sorry, 10,000 in Oxford. So 50,000 people at least have been vaccinated by one or other of these vaccines. And out of that 50,000 people, none have been admitted to hospital because of the vaccination, which I think is worth bearing in mind. 30,000 people in the study that Moderna has organized, with half of them getting the vaccine, half of them getting not having the vaccine itself. And there were 95 cases of COVID. Out of 30,000 people, 95 cases of COVID. In the vaccine group, so out of 15,000, 5 persons developed COVID. And in the control group, 90 persons. So almost, how much is that? I forgot my maths here. So I need my son at this point just to be able to show me what is what. So 18 times as many people in the control group developed COVID compared to those in the vaccinated group, with severe cases of COVID only occurring within the control group. And I think that is quite remarkable. In addition to this, Moderna are quite clear that they've had a varied cohort of people, with around 7,000 persons above the age of 65 in the total cohort. And they also vaccinated individuals who had chronic diseases, at least 5,000 persons. And significantly for us, 11,000 persons of color, 3,000 Black Americans. So a diverse ethnicity, diverse in terms of the underlying disease state and the age groups, suggesting that this is an effective vaccine in the highest risk groups. And we know that coronavirus is a virus that affects especially the elderly, and then those who have underlying heart conditions, who have been treated for malignancy, who have underlying lung conditions, diabetics, etc. And we talked about the fact that for the Pfizer vaccine, about 3.8% of persons who were vaccinated had fatigue, 2% had headache. And likewise, for the Oxford vaccine, in terms of reactions, the most common was fatigue and headache, with feverish and myalgia also noted. In the case of Moderna, the most common reactions actually occurred after the second dose. Now, remember, we talked about a prime boosting process. So you prime the immune system. You initially expose it, and then you get it to double down with the boost. And you expect a more vigorous reaction with that boosting. And at that point, 10% of persons complained of fatigue, 9% of muscle aches, about 5% of joint pains, 5% of headache, and 2% of persons had a bit of redness, all of which resolved. Like I said, no one was admitted to hospital as a result of this. No one required significant medical attention. In that context, what does that suggest? What that suggests to me is that when we come together with a massive collaboration with huge sums of money put in by government beforehand and afterwards in guaranteeing sales, Allah has given man the capacity to move mountains, and it is perfectly possible for him to do so. And that I think the due processes have actually been followed in this. It is a pity that that data and the publication of it hasn't come through transparent means, but the vaccines that are there on the books are effective and are safe. So where does this leave us in the UK? The JCVI, the advisory body advising on vaccination, has drawn up a hierarchy of groups who will receive the vaccine. And the top of this are the very elderly care home residents and workers in care homes. Looking at this data, certainly from Moderna and from Pfizer, they both suggest efficacy within this age group. And for most of us, which vaccine we choose and whether we are vaccinated will remain a theoretical matter until I suspect the middle of next year. We've received about 800,000, or will receive shortly 800,000, I think 800,000 doses of the Pfizer vaccine, which would mean that you could vaccinate 400,000 persons, which will quite rapidly be exhausted when we complete the care home vaccinations. And Pfizer anticipates being able to produce 1.2 billion doses next year. But that is global production. In that context, what would I advise? I would say that looking at this, these are safe vaccinations, and if we wish to protect those who are at highest risk, I would suggest vaccinating them with them. And on that point, I'd like to conclude. Let me just have a look at the questions that have come in, and feel free to ask any questions you may have. Muhammad will be uploading over the course of the next month short 10 to 15 minute snippets looking at in more detail at each of the questions that are here, both the individual vaccines and topics surrounding vaccination and COVID-19. So do subscribe to his channel and have a look at this as they come out.
Okay, thank you very much, Umar, for that very informative, thought-provoking talk. I'm just going to give you a minute if you want to drink some water or something. I just want to say a few words about a charity that's very close to my heart. As I'm sure you're all aware about the project we're doing in North Harrow called the Salam Centre, where we're trying to build a massive community centre open to everyone. And at the moment we have a Christmas challenge where we're trying to raise £12,500, and we have pledgers who have promised to double that if we manage to reach the target. So alhamdulillah, so far we've received £4,000, but 4,096. We still need to raise 8,404 to reach our target. So please donate whatever you can. You can find the donation link on the Salam Centre Facebook page. Hopefully it's open for another 2 days. So hopefully we can reach that 8,404 target over the next 48 hours. Okay, so Omar, if you're okay to take some questions. There have been quite a few questions that have come through. I've been trying to make a note of them. But first, I think, would you be able to clarify a few phrases you've used? So you mentioned mRNA. Is that the same as DNA? Is there a difference between mRNA and DNA?
So DNA is the genetic material. This is what's passed on from parent to child. It's what lives within that nucleus that we talked about. It's that vast library of protein, vast library that codes for protein. So imagine it's a huge library full of books that are tied together and kept in there. And every time that you need a protein made, a copy of that book is made, and that is copied as an RNA, mRNA, messenger RNA. That is then taken out of the nucleus, conveyed over to the place where proteins are made, the ribosome. They then read that book and produce the protein. So DNA is one thing, it's the material of inheritance. RNA is the material of life. It defines a living protein-producing cell. Your RNA will not be transformed into DNA and integrated into the nucleus.
Thank you very much. Okay, so moving on, just some practical issues regarding the vaccine, if that's okay. So for example, is the vaccine okay to be taken by everyone? Like, for example, if someone has severe COPD and have been advised not to take the flu vaccine, or someone currently has a cold, or somebody is pregnant, for example, can they all still take the vaccine?
So the trials that have been carried out looked at individuals who had stable underlying disease, including COPD, including heart disease, and showed that there was an immune response in those cases. So the patient who comes in with Mohammed, it's stable COPD or severe COPD, what we're looking at?
So the question asked, somebody said they only have 25% of function of their lungs left. They're very severe and they've been advised not to take the flu vaccine.
Okay.
So a severe case of COPD.
I find that interesting because the advice would normally be the other way around, that in an individual who has severe underlying respiratory disease and doesn't have respiratory reserve, so all of us have spare capacity. The less capacity you have in your lungs overall, the less spare capacity you have, the more likely if you become unwell that you will become very, very unwell. And someone who has 20% lung function, let's say, would be top of my list to have the flu vaccine unless they are heavily immunosuppressed. And even then, you would want to give them the flu vaccine if you could. So you'd be aiming to this is the cohort who would benefit from a coronavirus vaccine. Was that?
Yeah. Okay. What about pregnancy or somebody who currently has a cold?
So you don't normally vaccinate individuals who are unwell at the time. You wait until they get better in order to make sure that there isn't a confusion between a vaccine effect and their underlying, their current illness. So the person with a cold, you'd probably not vaccinate until they're over their cold. Pregnancy, none of these trials would have been carried out on pregnant individuals. And the highest risk groups we have are individuals at advanced age with significant extensive underlying medical conditions. So the kind of group that will not get pregnant. And they are the ones who will have it first. As we're seeing with flu vaccination, we are beginning to realize that pregnancy is a high risk group. For the pregnant are at high risk of respiratory viral infections when they get them, and should probably have the vaccines if we can get it to them. Current guidance on this is something that I'm not aware of, and we don't normally vaccinate pregnant individuals with the new vaccine. So I would need to look into this, and I can come back. If you leave your details with Mohammed, I can have a look at this and get back to you.
Okay, thank you very much. Okay, so just moving on to some of the data. Now, the data we've got some of them you mentioned have only followed people up for 1 month, maybe 6 months. How can we can say they're safe for a month, safe for 6 months, but how can we be certain about the long-term safetiness of these vaccines?
So all of these studies have plans for following people up for at least a year. So those who were in the study would be followed up well beyond their initial vaccination. We haven't reached a year since coronavirus became an issue. And you can, given the fact that in the context of, say, chimpadenovirus, when it's been, that vehicle, if you like, has been used in studying other conditions, long-term effects don't seem to be a problem in that context. So it's unlikely to be so here. When you look at cancer therapeutics and the work BioNTech has done on delivering mRNA to cancer patients, I need to look into this, but I'm not aware of long-term problems as a result of the therapy itself. So drawing an analogy from there, it's not likely that there will be long-term effects, but these are novel vaccines. These are things that have not been licensed on a large scale in human populations, and will require follow-up, and there will be follow-up. However, looking at the papers that are there, looking at the track record of the chimpadenovirus and previous therapeutic use of mRNA, that's less likely to be an issue.
And just going back to the people who can, you mentioned about when you talked about pregnancy and that, what about breastfeeding mothers? Because there was a question I think that's just coming on YouTube about the mRNA vaccine is a lipid emulsion. So what do you think about vaccinating breastfeeding mothers?
So what's the risk here? I'm not sure what the answer to this is, first of all, but let's think through this. What's the risk here? You vaccinate the breastfeeding mother with an mRNA vaccine that's surrounded in a lipid bilayer that's absorbed into her cells. And then it then produces the protein, and that's released from the cell. It's not released as a lipid bilayer that's then going to go into the breast milk. It's the protein that's released. And that protein is going to activate the mother's immune system. So I don't see a significant risk to the mother from that point of view. And the child, in absorbing that protein, is probably not at risk at all, actually, because their gut will digest the protein that's there. They're not going to be absorbing it as a protein, and consequently, the risk is not something that's significant. So I would not anticipate that breastfeeding mothers would be prevented from having the vaccine when it becomes more widely available. But I would need to look into the government guidance on this.
Okay, fine. Okay, so moving on now to some of the, again, more practical issues. If I were to have the vaccine, I believe I'd need 2 doses to get full immunity. But how long would my immunity last? And if I'm immune, am I still able to transmit the virus to other people? Or am I protected from doing that as well?
I think that's a very good question. Let me answer the second bit of it. So what do we know from the trial data that we have? We're looking at whether people develop the infection as a result of being vaccinated, whether they become infected. And what this data all shows is that they're not becoming infected. In order to be able to transmit, you need to be infected first of all. So it should break that chain of transmission and prevent spread within the community as a result. So after you're immune, once you're producing antibodies that recognize the spike protein on the coronavirus, what happens is that when you're exposed to the coronavirus, it's sprinkled with this magic dust of antibody, and your immune cells, your T cells, will come and consume that coronavirus. And consequently, you will not establish an infection and you will not be able to transmit to others. And I think that's important. The first part of the question, Mohammed, was...
So how long am I immune for if I've been vaccinated?
That is uncertain. That is uncertain. And we're going to have to wait and see. But where can we draw analogous data for this from? We can look at infection in individuals in the community currently who've then developed antibodies to the coronavirus as a result of the infection. There's a study from Oxford looking at healthcare workers and following them up for 6 months following infection and finding they don't develop subsequent recurrent infection. So for as long as they've been followed up, they've not had problems. That's about as much as we know at this point. We're not sure how long that immunity will last for, and it's something that will have to be revisited quite regularly.
Okay, thank you very much. Okay, so I've had a few more technical questions, if that's okay. So firstly, the trials you mentioned they were done on a mixture of healthy and unhealthy individuals, people of different ages and everything. So I think that's been answered already. But are the phase 2 and 3 data available for the public? And there was one question from a brother regarding the number needed to treat for the Pfizer vaccine. Apparently, from what I understand, this means that you have to vaccinate 256 people, and out of them, only one will really have a benefit because the NNT was 1 in 256. Is that accurate? Do you know anything about that, or are you able to shed more light?
I would defer to the epidemiologists on this in terms of working out the number needed to treat, but I suspect the number needed to treat is going to vary with the prevalence of the infection within the community and exposure to coronavirus. And the first part?
Other data, can we get phase 2 and 3 data? Is it available for the public? Or?
So the phase 2 and 3 studies that have been published are in the Lancet, where Maheshi Ramasamy is the lead author published on the 19th of November, Safety and Immunogenicity of ChAdOx1 nCoV-19 vaccine. So that's the UK cohort that's been looked at. So it's not the total group, which would be UK, Brazil, and US. That's just the UK data. That's there. Moderna has published in the New England Journal of Medicine. The phase 1, I'll dig it out for you here from what I've got. And that data, if you like, what's present within the papers is available. And the FDA, for instance, has pledged to release the data submitted by Pfizer in pursuit of licensing of the Pfizer-BioNTech vaccine. So that should be freely available as well.
Okay, there have been a few questions regarding the content of the vaccine. What is the vaccine made of? Are there any unhalal contents in the vaccine? And there have been a few questions regarding it containing stuff from aborted fetuses. Are you able to shed some more light on all of those?
So given that the vaccines have not yet been released, given that the SPC detailing the adjuvants and detailing its constituents has not been freely published, and given that that has not been published as is normal within the scientific press, I wonder where these claims are coming from. And the people who'd have clearer understanding of this would be the manufacturers themselves. I don't know what's exactly in them, and it's something I await to see. And some of the more lurid claims that are there about aborted fetuses being used in this massive industry dependent upon consuming children for the protection of the elderly. It's something that seems more like clickbait than reasonable concern.
Okay, thank you. There've been quite a lot of people have asked if I've already had COVID, can I still take the vaccine? Should I still take the vaccine?
So there are two different questions in that. Can I and should I? The trials were actually done at least within the Oxford cohorts within the 18 to 56 age group in individuals who were both seropositive, so who had the infection and had developed antibodies to it, and those who didn't. And I think the Moderna study also involved people who had or didn't select between those who had and didn't have the infection. So you can have the vaccine. There shouldn't be a problem. Originally, there were concerns about vaccine-related exacerbation of disease, where individuals might suffer from vaccination if they'd already had the infection. But this has not been shown to be the case in any of the trials. People who've had the infection have been part of it, and it's not been a problem with any other respiratory tract infection either. Should you have it? I guess that depends upon how effective is what we call wild-type immunity. How effective is immunity and how long-lasting is it after you've had the infection? That's an open question. Just as how long will this immunity from the vaccine last is an open question. And I guess I would look at it in terms of what is your risk of developing coronavirus? If you've already had the infection, you don't know how long the immunity lasts. That's the one concern. If you have significant underlying health concerns, it's probably better to have the vaccination in that kind of context. If you are elderly, it's probably better. So if you're falling into the high-risk groups, a counsel of safety would be to get vaccinated.
Okay, so you spoke about the 3 vaccines that are going to be available in the UK. There have been a few questions regarding the Russian and the Chinese vaccine as well, because apparently a lot of Africa, in a lot of Africa, these are the vaccines that are going to be offered. Do you know much of anything about it? Are you able to shed some light about these?
So the Russian vaccine is, in a sense, similar to the Oxford vaccine. It's based around another respiratory virus called adenovirus, but this is a human adenovirus and 2 different human adenoviruses, adenovirus 5 and adenovirus 26. It delivers a bit of DNA that's then made into RNA and used to produce proteins. All that I'm aware of is the very high level of reactogenicity with it, but I haven't seen the phase 2 or 3 data. I'm not sure whether they're published in the case of the Russian vaccine, and I certainly haven't seen it for the Sinovac, the Chinese vaccine. These 2 vaccines will be the subject of later discussion and some of the uploads onto Muhammad's channel over the course of the next month or 2.
Okay, thank you. And a few people have asked about people with autoimmune conditions. Can they still take the vaccine? People on immunosuppressants, can it exaggerate the symptoms of the original condition of arthritis or anything like that?
So people with immunosuppression are at high risk of coronavirus, and you'd normally recommend them to have the vaccine. People with underlying autoimmune conditions should probably also have the vaccine, particularly if they are immunosuppressed. Does it exacerbate underlying conditions? I would need to have a look at the more extensive data on this. It's not clear from the data published. Do we see this happening with other vaccines? Not particularly. So does vaccination itself per se exacerbate underlying autoimmune conditions? Not really. Will this kind of vaccination do that? I'm not sure. We need to get more data on this.
Okay, and now in terms of, so for example, there's a few people who own nursing homes and next week are going to be expected to roll out the vaccine to their staff. there's been very little information or anything given about the vaccine. So how do they encourage their staff to take the vaccine? And on the same note, if you were offered the vaccine tomorrow, would you take it?
So reading through the data that I have, I would happily take the vaccine tomorrow.
Is that all three, or is that which one?
Are you suggesting all three in one, or is it?
No, as in if you were offered, as in would you take whichever one you're offered, or would...
So I would be happy taking whichever one I was offered on that site. I can understand the concerns here with regards to efficacy. All of them have reasonable efficacy. The Moderna and the BioNTech vaccines appear to have a higher efficacy. We're waiting at least some more data from the AstraZeneca teams with regards to their low-dose vaccine, which seem to have a higher efficacy.
So you mentioned efficacy a lot. Is that the same as effectiveness? What does efficacy mean?
So efficacy here is within the trial context, how protective the vaccine is. How many people are likely to be protected from developing an infection once they have the vaccine, or what percentage? So I don't have concerns about either of those vaccines that we have here. And
how do we encourage our staff to take it?
So the key behind this, I think, is whether you feel comfortable taking it yourself, having heard what you've heard now. And staff having enough information to make a sensible and reasonable judgment, and understanding why they are being asked to take the vaccine. The reason here is that they're dealing with an age group in which 10 to 15% of people who develop the infection will die of it. In the over 80s, it's not a minor matter. And you don't want to be the person who's transmitting it to them. So protecting is a matter of protecting both the staff body and also the patients they're looking after. And if anyone wants to approach me afterwards, I'd be happy to discuss this on a smaller scale with individual staff groups, so that we can see what their concerns are and address them appropriately. And I think the important thing here is to work out what people are worried by and what they're worried about, because the national response, unfortunately, has not really engendered trust. In fact, has caused a lack of trust within what's been happening.
Okay, I'm gonna, Ahmed Versi wants to ask a question personally, if you don't mind. So he's gonna, Ahmed Uncle, are you happy to ask?
Assalamualaikum.
Right.
If a person who's got already had a vaccine and is immune, and then that person meets another person who has COVID and has not had the vaccine, and then the guy sneezes or whatever, and then I inhale. I mean, the person who's had vaccine inhales the person's droplets. Apparently, because the droplet stays in the nasal passages, in the nose, of course, the person who's got the vaccine is immune. However, that person who has inhaled it, is he able to then transmit the COVID to another person who doesn't have a COVID? Because he's still got the COVID in his nasal, because the vaccination does not kill the COVID in the nose, right?
So I think we need to address this quite carefully here. How is it that this virus is spread? It's spread by the release of virus from a person who is infected with it in their breath. It's not spread in the breath of someone who happens to breathe in the virus. These are the products of a febrile mind concerned about minutiae on this. I would not worry about that because it's not transmitted that way. The vaccine will not kill the virus that rests on the nasal mucosa, but any antibody that's produced will also neutralize the virus. And in any case, the amount of virus that comes from an individual into someone else's nose is tiny. It's not then generally rebreathed out. What is breathed out is your own respiratory droplets, which are far more in number. So I wouldn't worry about that. There is not a risk of transmission in that context. Does that answer your question, Uncle Ahmed?
So yes, I have understood, but I think one of these scientists did mention this in one of the discussions, that this is a possibility of transmission. So I don't know, I'll have,
I would invite them to come on to a ward where there are actually coronavirus patients so that we can discuss this then.
Okay.
Thank you, Umar. Okay, so just something about the approval process in the UK. Would you be able to shed some light on why it's been approved by the MHRA, but other agencies like EMA and FDA aren't ready to approve it just yet?
So the inner workings of the MHRA are a mystery to me. But what has happened is that because of Brexit and the inevitable grand wisdom of the powers that be, they've had to resurrect the MHRA before the end of the year in order to carry out this approval process. Whether the MHRA has been leaned on to fulfill this before it's had an appropriate chance to review the data was a question that's been raised. But even in that context, look at it this way. You have phase 1 studies, we have phase 2 and phase 3 studies, and a rolling process of assessment from the MHRA where they've been working with Pfizer for a bit, looking at the data. We have published data that suggests, at least with Moderna, and early published data in the case of Pfizer, but not the summary, which suggests reasonably good efficacy and safety. So from a safety point of view, I'm not worried. I am worried from a trust point of view, and I am worried that the MHRA has not set the standards of transparency that are needed in order to win people's trust. And that this has been turned in the comments of some rather childlike ministers to a school ground game to show that the UK is better than other countries for a vaccine that's produced by a Turkish pair living in Germany, manufactured in Belgium, trucked across France, and then ends up in the UK without an iota or an involvement in the process. So in short, I'm not sure why the MHRA has decided to do what it's done. My only real concern here is that it hasn't followed, or it hasn't set the precedent of transparent, good medicine with the release of all data that it has at hand.
Okay. So somebody's just coming back to your answer from Ahmed Uncles. Just saying, then why do masks need to cover the nose as well as the mouth?
Okay, so who is being protected by the mask? I think that's the key question here. When you're breathing, most of us breathe through the nose as well as the mouth. Some of us are primarily mouth breathers, but most of us will breathe through nose and mouth. And whenever you're breathing, you're going to be releasing these droplets that come off the wet membranes in your mouth and nose, and these will contain the virus itself. So it's only by covering the nose that you're going to catch these droplets in the mask and prevent other people breathing them in sufficiently. So Ahmad Angel's question was whether an individual who is immune breathes this in, when they breathe out again, will the tiny amount of droplets that might have lodged in their nose and might then be breathed out be a source of infection? And I think, no, that's not the case.
Okay, so just trying to get through all the questions now. So there's a few quick ones. So firstly, is it true that the studies ignored symptoms or side effects until 42 days after the first dose of the vaccine?
So as I read out to you, all of the participants in these trials had diaries to keep for 7 days after each of their vaccinations, documenting even the most minor complications that they may have. And individuals were followed up reasonably frequently, at least 8, in some cases, at least 8 times over a 2-month period following the initial vaccination. So no, it's not just after 42 days.
Okay, is there any indication of cross-immunity from other coronaviruses, such as SARS and MERS, arising from these vaccines? Okay, so will you get immunity to SARS? Will you be immune to MERS or SARS as well if you're having the coronavirus vaccine?
Right. The only way you would really know that is by having people challenged by MERS or SARS, and I don't think that would be approved by any ethics committee.
Okay, and everyone talks about over 80s being a higher risk, but is there really a difference between people age 79 and 80 or 77 and 80?
So the highest risk is going to be in those who are over 80, but risk starts increasing from the age of 65, and it increases quite considerably beyond with each step beyond that. But this is just the way we've collected the data.
Okay, and what about the immune system recognizing our cells that are producing the viral protein as foreign and destroying these cells? Is that a concern?
So when a cell is infected, there is a more extensive derangement of its function. There are more proteins that are produced, and the virus itself induces destruction of these miniature organs within the cell. It's that kind of cell, the infected cell, that the immune system tends to target and destroy. All cells within your body have got receptors or molecules where they display proteins that are being produced within them or that are present, digest them and display them on the surface. These, once they're recognized as foreign, would induce a immune response. An isolated protein being produced on the surface need not necessarily induce destruction of the cell.
So I'm going to ask my own question now, if that's okay. So the vaccine that we're going to be offered in hospitals is most likely to be the Pfizer vaccine. Now, the Pfizer vaccine has to be stored, from my understanding, at -90 degrees centigrade, and it has to be given within 4 days of defrosting. And when it's defrosted out here, it's very unstable. So you can, in terms of transporting it in anything with wheels or something, apparently destabilizes this vaccine. So what is the likelihood that when I am vaccinated by the Pfizer vaccine, it's still active and still working, and it hasn't been destabilized en route from the freezer to me?
So I'm not aware of the extent of its fragility, but mRNA vaccines are fragile, and this is part of the reason why they're being stored at such low temperatures and why such great precaution is taken over the defrosting, the storage within the fridge, and the administration. In a hospital setting, hospitals have got fridges that are capable of storing this, and it will stand being in a fridge for 5 days. So I don't anticipate the efficacy of the vaccine, the effectiveness of the vaccine that you receive, being any less than that the trialists have received.
Okay, but do you see a problem with distributing the vaccine across the country, even across the world, given its need to be kept at -90 degrees?
So yes, absolutely. I mean, this is the greatest challenge that's there. In advanced healthcare settings like the US, the UK, Japan, Germany, etc., where you have extensive public health networks, you have cold storage chains that can be maintained. This is not going to be a problem. But this is going to be nigh on impossible in the majority of the world where we cannot maintain that kind of cold chain. And this is actually where the ability to keep the vaccine between 2 and 8 degrees for up to a month is going to be more useful.
Okay. Okay, I think we've been going for 1 hour, 20 minutes now. I think we should probably call it a day. Thank you very much.
I'm happy to continue as long as you wish, Mohammed.
You're happy to? Okay, fine. Okay, we'll have one last question because one's just coming on YouTube, if that's okay.
Fine.
So looking ahead at the biotech firm's infrastructure of vaccine creation, does it have greater adaptive capacity now than pre-COVID because of all the mobilization of resources, given that there are likely to be more of these things kicking off going forwards?
So given the ecosystem that we've created, the ability of governments to invest vast sums for corporations. And what's interesting here is that of the 3 vaccines that are there, all of them are from challenger firms. Moderna is a firm that's outside of the traditional vaccine industry, as is BioNTech. And BioNTech has teamed up with Pfizer in order to scale up production rather than initiate it. The AstraZeneca vaccine is actually an Oxford vaccine. It's an academic vaccine. It comes from that Oxford vaccine group legacy, if you like, and AstraZeneca is scaling it up. So these are not coming from Big Pharma originally. Big Pharma is involved in the scale up. Government has put in huge sums into this, both from the research, developing it in the background, and in the purchase of vaccine before licensing, and also in investment. So it's shown the capacity of government to drive this. It's shown the capacity of education institutions to produce this. What the current setup hasn't shown is the capacity of public sector stakeholders to demand a fair and just cost and distribution of this. And I think that needs to be a precedent that we need to set. But yes, the ecosystem has changed. There are new ways of reducing risk. There is a new capacity for larger bodies, government bodies, to accept that risk. And yes, you're right. This challenge that we're facing today is the worst of a series of pandemics that have occurred over the last 30 years. Every 10 years, we've had a coronavirus pandemic or a flu-related pandemic. This is the worst that we've had in living memory, but there will be more. And it's a matter of how we adapt to deal with that challenge that's important.
Okay, so last question then. Where do you see us in 6 months and 12 months' time? Do you still see us locked down in our houses? Or do you think we are going to go back to what we consider normal?
I worry that the hunger to return to normality is a failure to recognize just how dangerous that normality was. And that in everything there is a hikmah, in everything there is something for us to learn. And recognizing that the broken world that we had created before is not the world that we need to grow is something important. And I hope that we don't return back to normality. But I hope that we are able finally to return to one another's company. And I see that happening. I don't know. I don't have a glass bowl here, but I don't see that happening in 2021. Primarily because of the following. We will need a clear and sustained reduction of transmission of coronavirus within the community, a high level of vaccine uptake within the community, in combination over a prolonged period of time before we are able to dispense or to retire the hand washing, the social distancing, and the mask wearing that's governed our lives for the past few months. And the trigger point with regards to the prevalence of coronavirus in the community is something that I'm not too clear on, but I don't see that being achieved at any point during the next 12 months.
Okay, thank you very much. I think we've asked all the questions that have been posed, or as many of them as possible. Thank you very much for your time. We will try and put something together over the next month on the YouTube channel. Please subscribe so that you know when they come up. And inshallah, we'll arrange further sessions in the future, depending on what the issues of the day are.
Yeah, alhamdulillah, for me, this has been a remarkable and beneficial learning experience, an opportunity to review things that you take for granted, and I am grateful for that. So, jazakumullahu khair.